Thiamethoxam and Clothianidin insecticides lower the Apis mellifera queen's reproductive rate. Presented by Courtney Kramer. First, I would like to introduce to you the western honeybee. The genus is Apis, meaning bee, and species mellifera, meaning honey-bearing. The origin is believed to be from Asia and Africa. This species functions as a superorganism, and it is a highly developed eusocial class system. It is important because this species is our primary pollinator for plants around the globe. Now I would like to talk about the basic reproductive process within Apis mellifera. It has a class that is solely devoted to reproduction. The primary member of this class is the queen, because she is the only one who has advanced reproductive organs, as displayed in this picture. The queen practices extreme polyandry. This means that one queen per hive mates with many males to produce offspring. Now I would like to talk about the insecticides, Thiamethoxam and Clothianidin. They were first seen in the mid-90s. They are neuroactive insecticides. They are chemically similar to nicotine. They are also water-soluble, which is interesting because if they are laced on a seed, they can be absorbed into the vascular system of a plant. This means that if a bee or any other insect lands on the plant, it'll die. They're really effective. This is why they are used widely around the globe, but they can also pose a great danger to honeybees. Does an Apis mellifera queen's reproductive capability decline following the exposure to Thiamethoxam and Clothianidin insecticides? A study was done researching the effect of these insecticides and if they reduce the number of Apis mellifera queens laying eggs and the life of those eggs at 4 weeks. This experiment exposed developing honeybee queens to Thiamethoxam and Clothianidin to concentrates expected in the wild. They allowed the queens to sexually mature and then observed the production of worker offspring for 4 weeks. Figure A from the experiment demonstrates that there was a 38% decrease in queen-produced workers' laid eggs. Figure B shows that there was a 34% decrease. Fewer queens were alive and successfully producing worker offspring at 4 weeks. In conclusion, these insecticides severely reduced the number of Apis mellifera queens laying eggs as well as the number of queens still alive and reproducing at 4 weeks, thus causing a reduction in the number of viable queens. This same experiment studied if these insecticides reduced stored spermatozoa and viable sperm in Apis mellifera queens. Figure B shows that there was a 20% decrease in stored spermatozoa count, and Figure C shows that there was a 9% decrease in proportion of living sperm versus dead compared to the control groups. These insecticides significantly reduced the quality of stored spermatozoa and the proportion of living sperm in Apis mellifera queens, thus reducing the queen's reproductive viability. The same experiment also studied if these insecticides caused a change in the flight duration or mating patterns of the queens. Figure A and B show that there was no significant change in flight duration compared to the control groups, and Figure C demonstrates that there was no significant change in mating signs compared to the control groups. These insecticides did not alter the number of duration of flights or the number of mating signs in Apis mellifera queens. From outward appearances, the queen's lost viability may go unnoticed to human observers. In conclusion, this important study by William et al. Demonstrates the effects caused by the neonicotinoid in pesticides. The effects include a reduction in the number of queens laying eggs and the number of queens still alive at 4 weeks, the number of spermatozoa, and the number of sperm alive. Interestingly, these insecticides do not cause outward signs of distress, such as changes in flight quality or duration or even in mating signs. Agricultural use of these neonicotinoid pesticides may result in a negative effect and could be a decline in honeybee population. The queen's decline in reproductive health is not obvious. This may lead to skepticism regarding the significance of the negative effects of these insecticides. A decline in a wild population of Apis mellifera and decreased population around the globe may result. Lastly, a serious consequence could be seen in less quality crop production for food security. The effects of these insecticides would be brought to the attention of the public and private sector leaders because the fate of our growing world population relies on the health and goods honeybees provide through participation in the ecosystem as primary pollinators. These are the references I used to create this presentation. Thank you.